in,
Is that okay, Alex, by the way?
oh yeah,
We
for
usually
sure.
post all these. Great, great. Okay.
Yeah, no problem. All right, yeah, thanks everyone, and thanks Carlo for hosting. And so in my training, I was always motivated and interested in this question of brain expansion. And I focused on genetic, molecular, and cellular mechanisms that might drive these dramatic changes in brain structure and function that evolved along the lineage leading to humans. But one of the special properties of mammalian brain expansion is that as brains expand, not all regions scale equally. There's this process of allometric scaling, such that the neocortex, underlying white matter, and striatum expand disproportionately compared with regions such as the olfactory bulb or ventral midbrain. And we became interested in how this process of unequal scaling, combined with new functional requirements, may drive cell type specific adaptations to our large brain cellular ecology. And we think that these trade-offs, especially between the size of target regions and source neurons, may be most strongly manifest in the ventral midbrain. Here, a small cadre of a few hundred thousand hardworking dopamine-producing neurons project to target regions in striatum and cortex that have expanded disproportionately. And we think this may represent a trade-off in brain expansion and one of the costs of brain expansion. Um, with collaborators, we measured the volume of these target regions and can see compared with old world monkeys, they've expanded 6 to 17 fold in humans, compared with only a two-fold expansion in the number of source neurons. And then, as I alluded to, Mary Rigonti's group measured TH innervation in a number of regions. And so not only has the volume these neurons project to increased, but the innervation density has also increased. Here showing medial caudate comparing human even with chimpanzee. And there's this dopamine-directed striatum hypothesis that this increased innervation may have actually been important for the evolution of prosocial and flexible behaviors. So we have both unequal scaling and potentially new functional requirements, increasing the demands on dopaminergic neurons. And these neurons themselves have extraordinary specializations. The axonal arbors are largely unmyelinated and can span over a meter from a single neuron, even in rodents. And as such, they have high bioenergetic demands, particularly in nigrostriatal dopamine neurons that maintain pacemaker activity, which is an energetically expensive process. And the production of dopamine itself releases reactive oxygen species, further making these neurons vulnerable. In addition, it was reported that the human and chimpanzee dopamine neurons have this coiled morphology not seen in other species, which would further increase the branch length. As such, these neurons are implicated in a number of disorders enriched in humans, most notably Parkinson's disease caused by their loss. And so we became interested in this question of whether dopaminergic neurons have evolved in response to these trade-offs related to brain expansion, and whether we could investigate the developmental origins of connectivity differences, as well as whether there might be compensatory adaptations that allow these neurons to inhabit our large brain cellular ecology. And to look at this, we were fortunate to have Sarah Nolbrandt join the lab from Mullen Parmer's lab. And she brought with her an excellent protocol going into clinical trials for generating human dopaminergic neurons in 2D. Sarah adapted this to 3D protocols and to work with the common protocol across different primate species. Um, but we wanted to go a step further and study many individuals in human and chimpanzee to identify species differences as distinct from individual differences. And to do that, we wanted to do this in an efficient way where we could also mitigate batch effects. So we set up this village in a dish culture approach with intraspecies pools where we pooled together either human or chimpanzee, exposed the cells to dual SMAD inhibition, sonic hedgehog for ventralization, and activated Wnt signaling. And we bracketed this such that every line, since lines vary in their response to Wnt, would receive appropriate Wnt in some of the conditions for caudalizing. But in addition, we wanted to isolate cell intrinsic species differences. So Sarah also set up these phylogeny in a dish culture conditions where we pooled as many as 17 individuals from 4 species in the same dish at the pluripotent stem cell stage, and then used the same protocol to generate these interspecies organoids. Here's an example of what the experiment looks like. Here, Sarah thought out IPS lines from 17 individuals across 4 species, patterned them for 16 days, and we could see consistent expression of these ventral midbrain markers. Um, and we use these different shear concentrations, which would be expected to increasingly cauterize the samples. And indeed, when we did single cell RNA-seq at this stage, we could see a gradient of cells organized by the quantity of cheer, this Wnt agonist they were exposed to. And this matched gene expression patterns from the caudal diencephalon to the midbrain hindbrain boundary, with a plurality of cells representing ventral midbrain. And we can come back to this in discussion, but there were a few strategies Sarah took methodologically to employ this large-scale multi-individual culture, and that involved quality control of the lines up front, removing lines with p53 mutations, and pilot differentiations on many of the individuals. And I have a few other strategies I can mention later. Um, we could then cryopreserve these 2D cells, thaw them out into 3D spheroids, and grow these in culture where we would see, as Ron anticipated, the TH and FOXA2 expression with TH positive fibers on the outside, indicative of dopaminergic neuron processes. And we can maintain these for long term in culture here out to 100 days. We could even do this for 6 months. And we then wanted to assess what is the reproducibility, fidelity, timing, and maturation of these mixed species organoids. Are we on target? Are we preserving all of the individuals in this pooled culture format? And to look at that, I'll have to take you on a detour with this informatics tool that Nathan Schaefer in the lab developed. Um, so we can use the sequence variation in transcripts to assign cells to their species and individual of origin. And when we started, I thought there would be off-the-shelf tools to enable this. But we encountered a number of complexities with this experimental design, as well as opportunities to improve demultiplexing. And so first, typically people would do these barnyard experiments just to check for doublets with new single cell technologies. And they would align reads to a composite genome of multiple species. But this becomes computationally prohibitive once you get up to 3 or 4 species and is very slow, even with 2 species. So Nathan developed a more rapid reference-free alignment method, demux species here, where he could pull out species-specific strings of sequences or k-mers and quantify these to assign cells to species of origin. And then, while there were existing tools for assigning cells to their individual of origin, these tools were slow to run, and they also sometimes, at least we anticipated, would encounter problems with deep population structure, like we might see in subspecies of chimpanzees. And so Nathan made a new tool that would consider different sets of SNPs for different comparisons and run more quickly, called demux VCF. Um, we also wanted an independent measure of the individual of origin and one that could run alignment free. For example, if you're setting up a big pooled experiment, what if some lines drop out, or if you have a contaminating line you didn't anticipate? And so Nathan developed a method for determining the mitochondrial haplotypes of origin in each individual cell. And this method can even run with demux VCF if you don't have variants to begin with to determine the individual variants. And in addition, you may want to have tags in your experiments, like we tag different levels of Wnt perturbations. And he developed a new method for recovering these tags even in low count cells that does not just employ population averages to be more sensitive. And one of the things I think we've all encountered who've done single cell RNA sequencing is that when you process cells and dissociate them, it lyses some of the cells in nuclei and releases ambient RNA. And there's existing methods that are quite good at identifying the identity of ambient RNA transcripts as a whole using the empty droplets. or using heterogeneity between clusters. But these methods disagree with each other at the per cell ambient RNA level, making it difficult to properly correct for ambient RNA. And so Nathan realized that when a variant and a transcript mismatches the predominant variant in the droplet, it's a ground truth for ambient RNA. And he used this external ground truth for a new ambient RNA removal method that we've applied here. And then finally, he has another tool called Doublet Dragon that can harmonize the different doublet rates reported by these different methods to produce a global doublet rate that can be useful in downstream analyses. And I'm not showing it here, but he also has a method if you wanted to do pooled culture and then bulk RNA sequencing, kind of like census-seq for pulling out the composition of individuals. And so you could use this bulk proportions method as a quick assay, low cost assay for determining whether you've maintained your individual proportions before going to single cell RNA-seq. And so we call this cell bouncer for checking IDs and keeping the riffraff out of single cell experiments. There's a preprint available, and Nathan actually put this to the test on the hardest demultiplexing method we could think of. We've been growing these human chimpanzee tetraploid cell fusions in the lab that contain the genomes of two individuals per line, as well as three different possible mitochondrial outcomes. And using the combination of tools that Nathan developed, he could identify the combination of donors from each cell fusion, as well as whether they maintain mitochondria from just human, from both species, or just chimpanzee. And predominantly, the human chimpanzee fusions maintain just the human mitochondria, but he could also find rare exceptions of cells that matched these other cases. And then he could use these to see what's different about these cells. And he found evidence for both mitochondrial incompatibilities as well as nuclear mitochondrial incompatibilities that you can read about in the manuscript. But returning to the dopaminergic neurons, now using Cell Bouncer, we could pull out the single cell gene expression profiles as well as the individuals of origin. And we could see that we recovered all individuals in the experiment at reasonably comparable proportions, especially for human and chimpanzee, and that about a third of the cells represent the LMX1A dopaminergic neuron lineage. So we have on-target cells from all species and all individuals for downstream comparisons. Now, how reproducible is the assay? Um, we can look
Hey,
at
Alex, could
every,
I could I
yeah,
get you
please,
to rewind one
let's do
slide?
it. Yeah.
So, I think one of my surprises actually when I saw this figure or a similar one in your paper was how well you get line representation. There's not a big purple bar with a bunch of tiny little blue ones. You have a really good number of cells from most of these lines, and you're not only doing it with 5 human lines, you're doing it with many interspecies lines. Is there anything, I guess I would say, qualitatively different about how and when you're mixing and growing lines than other groups? I think most groups would say that when they try to mix them early, they get a line that takes over or 2 lines that take over.
Yeah, I
Is
think
that
this is kind of the crux of why we're meeting for village culture methods. And when I was a postdoc, I did some of this mixed species culture and
oh
assayed it with the Fluidigm system. And we would have different individuals grow at different rates in cortical organoids. And we'd only get like 50 cells on a chip. So I'd have like 40 human cells and like 8 chimp cells. So it wasn't that useful. And I had a rotation project, and we did find that mixing lines for cortical organoids, we would experience kind of this compounding effect of different growth rates, such that in longer term expansions, one individual might be more likely to win out. And you can see in some of the published studies, the pre-patterning is done separately, and then the lines are mixed at the neural progenitor cell stage. And you lose a lot of the efficiencies if you have to culture, 20 lines
right,
separately
right.
for several months. And so what are the things that we did differently? I think one of the things is it's easier with the ventral midbrain because neurogenesis happens earlier. So you don't have as much of the compounding effects. Another thing, though, Saro was very careful about the IPS culture conditions and maintenance. Every line was cryopreserved and banked in the same state in the same way before starting the experiments. And we ruled out lines that had P53 or other mutations. We also, in one of the experiments, we did have an experience where one of the human individuals came to partially dominate. But that happened between day 40 and day 100. And so we modified the protocol to add a gamma secretase inhibitor at day 40, inhibiting Notch signaling that would promote cell cycle exit. And that solved the problem for that one line. And so I might recommend a soft manipulation, like promoting cell cycle exit when your cells have reached the stage of neurogenesis, as well as just a lot of like QC and consistent IPS culture going into the experiment. And when you start the experiment, also the processing really matters. So if you're trying to dissociate 20 lines in sequence, if some lines are sitting dissociated longer than others, they will suffer. So you have to really game plan out how you're going to do it. Maybe have multiple people there for pooling day, so that you can parallelize that one aspect. But then after that, it's one organoid. And, I appreciate your positivity here. I think it was a real challenge to get this kind of representation across individuals. And so proud of the work Sarah did here. I think in our initial pools, we only had one orangutan and one rhesus macaque individual, and we seeded them at a low fraction to start. And I will say that, there was a reviewer who felt very strongly that the low fraction of those cells affects the overall experiment. And so it
That
is,
it's hmm,
there's always a glass half empty, half full perspective. But, returning to the evidence why we think the glass is half full, even beyond the pooling, is that if we compare all these different pool types, the within species and the cross species pools across all these different individuals, we can see really comparable, not just representation, but also cell type composition across both lines and pool types. And for further quantitative analysis, what's so beautiful about these pooled culture experiments is you have so many levels of experimental design where you can assay reproducibility. So we can look at the correlation of marker gene expression for cell types across individuals, across pool types, across experimental replicates, across different shear concentrations, or across species. And we can see a very tight correlation. When we make cells of the same type, they have very correlated marker gene expression. And you can see that's greater for all the within species comparisons than for the cross species comparisons. And we think this reflects the real biological differences between species that we're powered to detect for having such tight within species correlations. In addition, we can assay the fidelity by mapping two developmental brain atlases. But it's a real challenge when you're outside of cortex because there aren't that many cells in the brain atlases. And so we could see very high fidelity that the cells we assign in our study as dopaminergic neurons map to the DA lineage in the Linnerson atlas. We also, I'm not showing it here, but we tried to make our own atlas of rhesus macaque midbrain development and collected a smaller number of cells since it's such a rare population, but we could see a similar fidelity. And we can see, just as an example, the shared marker gene expression for dopaminergic neurons across all species with reference to the primary organoids. We can also try to look at the maturation or match the age of these cells. The problem is the atlases are incomplete and they kind of stop mainly at day 100. We added a few older cells from later stages, but the whole cell dissociations make it tough to get DNA neurons out. And what we could see is that the older our organoids get, the more likely the cells were to be nearby the oldest cells in the atlases. But it's hard to put an absolute maximum date because there aren't many older cells in the atlas. But we do see ongoing maturation in the organoids through day 100. Um, the cells themselves, if we make an assembloid, the DA neurons can generate processes that will project into target regions like a forebrain organoid, and they have spontaneous activity and are capable of dopamine release. We can try to more quantitatively assess maturation using the pseudotime methods, and we can see an expected sequence of gene expression changes over pseudotime, matching dopaminergic neuron maturation. And one of the things we noted was that the rhesus macaque DA neurons mature much more quickly than the other species. So at day 40, you have a subset of the DA neurons that are comparable to day 80 in the apes. The human and chimp were very similar by another of measures. Maybe you could argue there is a very slight delay in human, but regardless, we can use this pseudo time trajectory as a covariate in the linear mixed models to account for maturation differences between species. And as an orthogonal measure of maturation, we can look at the switch from neurogenesis to gliogenesis, and we can see that at the same stage where there's very few astrocytes in the apes, the macaque has already switched to a great deal of gliogenesis. And this may be why macaque is underrepresented in our pools. We only had a small number of individuals, but it could have been this earlier switch out of neurogenesis. So then Janelle Wallace in the lab for these quantitative comparisons of gene expression between species led the analysis implementing this Dreamlet precision-weighted linear mixed model for accounting for the complex experimental design and finding the extent to which a gene's variation is explained by species as opposed to these other experimental parameters. And Janelle found a subset of cell type specific changes as well as constitutive changes and running this whole gamut. And she also used the outgroups to polarize these species differences to identify those likely to be human specific, derived in our lineage. And you can see one example here. We went on to validate this inward rectifying potassium channel, showed very specific expression in the human dopaminergic lineage. And we intentionally picked an organoid that had a mixture of dopaminergic and non-dopaminergic identity. So you could see the specificity, and we couldn't detect any expression of this with a perfectly matching probe in chimpanzee or rhesus macaque. And this was concordant with the differences in chromatin accessibility, where we saw derived human-specific accessibility at the promoter. And so, went on to find a more broad set of differentially expressed genes with concordant accessibility changes. And here's one of our favorite candidates, this neuritin-1 gene, which is a neurotrophic factor that promotes axon outgrowth and other cell types, which has 3 or 4 concordant upregulated peaks or peaks with increased accessibility in human, as well as derived quantitative upregulation in human. Um, Intersecting with sequence variation that might be causative of such dysregulatory changes, we found that structural variants were enriched for overlap with differential accessibility. And here's one example of a human insertion near a peak that is derived reduced in humans. And we could see that's concordant with a derived reduction in the expression of this GABA receptor subunit. We also sought to look for candidate trans regulatory changes. Jing Wen in the lab used CNIC Plus to identify regulons, correlations between transcription factors, the accessibility of their motifs, and the expression of their target genes. And in general, these regulons were correlated between species in terms of their number of target genes, target regions, specificity of target genes, etc. But we looked to see whether there were regulons enriched for being upstream of species differences in gene expression, like ZFHX3 or UNCAX. And we could see this ZFHX3 as a candidate transregulator of species differences that itself had a derived quantitative upregulation in humans and was also upstream of an enriched number of differentially expressed genes. Um, when I looked at the overall categories of differentially expressed genes, coming back to our hypotheses, the top ranked category in immature neurons related to axonal transport of mitochondria, including some ZFHX3 targets, and gave us insights into candidate genes that might be involved with early connectivity differences for further studies. What we were most excited about is the top ranked category of differentially expressed genes in the more mature dopaminergic neurons related to oxidative stress buffering. And this included a gene catalase that was recently shown to have a derived upregulated gene expression pattern in human cortical glutamatergic neurons. We saw that same pattern in dopaminergic neurons, as well as derived upregulation of many of the peroxyredoxins and other genes in this pathway. And coming back to our hypothesis, this suggested to us that even at baseline conditions earlier in development, there might have been evolution of increased buffering of reactive oxygen species. And so, to further test this hypothesis, we then went on to treat these mixed species organoids with rotenone to disrupt the electron transport chain. And I think this is an example of how powerful the pooled culture methods are. It's very hard to culture organoids from different individuals or different species in common conditions. The organoids are different sizes, they're exhausting the media differently, they have different size necrotic cores, etc. But by putting all of the cells in this common garden environment, we could directly compare them while mitigating these batch effects in their response to a common perturbation. And when we did that, Sara optimized conditions such that by 72 hours of rotenone treatment, we could recapitulate the selective vulnerability of these dopaminergic neurons. And we looked for the major axes of transcriptional variation. In general, we saw this overall conserved transcriptomic response. The major axes of variation relate to a shared trajectory of stress response and to species differences. And we could see that reflected in the expected upregulation of early response genes, heat shock proteins, and other metabolic genes, and the downregulation of some core neuronal markers. But we could also see species-specific differentially expressed genes that were unmasked by these perturbations. And these included a number of responses, like a human-specific upregulation of BDNF and downregulation of this mitochondrial calcium uniporter, that were shown to be neuroprotective for dopamine neurons in other contexts in the same direction. And then more generally, if we look at this stress response, although they share this conserved trajectory, we wanted to evaluate whether the baseline or stimulus-dependent response neuroprotective factors might change the overall extent to which cells traverse on this stress response. And indeed, across all seven chimpanzee individuals versus all 8 humans, we saw this pretty big difference in 24 hours where with the same stimulation, the chimpanzee cells responded more. And we could also see that still reflected at 72 hours. And so to conclude, we established this phylogeny and a dish approach for studying the evolution of human dopamine neurons. Um, we could identify candidate cis and trans regulatory changes that might underlie gene expression divergence and produce a resource of gene expression and chromatin accessibility during normal dopaminergic neuron development. And we could find, even at baseline, a human-specific increase in genes regulating axonal transport and oxidative stress. And we could unmask further candidate neuroprotective factors in humans, as well as some evidence for this hypothesis that there is, in fact, increased buffering in the human neurons that might relate back to a response to the increased demands and unequal scaling in our brain evolution. And in the future, we'd love to take this model further and also look at cell extrinsic factors related to oxidative stress and to more directly probe connectivity. And so this was part of a big team project led by Sarah Nolbrandt, Janelle Wallace, and Jingwen Ding for the organoids, and then Nathan Schaefer and Brian Pavlovich led the cell bouncer and tetraploid work. And here is our Halloween dopamine neuron that Janelle made. And I will stop there for discussion.
Thanks so much, Alex. I think there'll be a lot of questions, or I have a lot of questions anyway. Who wants to jump in? Anybody? One of the first things I was wondering is that the axes along which you're seeing species vary, do you think those are going to, or have you looked if, because I think even built into your experiment, you could look at this, the axis, let's say an axis that separates chimpanzee from human, is that going to be an axis that separates humans as well? Kind of how far along any particular human genetic makeup has pushed itself along this axis, or does it look like there's a whole different game when it gets to be variation within humans?
Oh, yes. So that's a great question. So in general, if you do some kind of simple analysis like PCA to find the major sources of variation, you'll get a distinct component that relates to species. It's a strong source of variation. And some of that is due to alignments and assemblies, which we've been trying to correct for. Nathan has been using this comparative annotation toolkit and has built this cat toolkit and has built another tool called Litter Box to clean up the annotations. And so we've done that to try to look at biological differences. In general, this component related to species has limited individual variation.
Okay.
Um, that being said, um, if we looked at the differentially expressed genes, the interaction between rotenone and species, we found that the differentially expressed genes that follow this pattern, the chimpanzee responded more to the rotenone for a given stimulus than the human did. So if we looked at the effects of stimulus alone, we could see that that variation was also further reflected between species, which was supportive of this increased buffering capacity hypothesis. Um, but I do think it's interesting. I always worry that, about individual variation in iPS cell experiments, that it reflects other anomalies with the stem cell lines. And if we see something like that, it might reflect anomalies between species as well as between individuals. We always want to be careful. There's the, skeletons in the closet we know about, like P53 that we can rule out, but presumably there's other adaptations to cell culture. And to me, that's why these village culture experiments are so important, so we can include a sufficient number of individuals to account for individual variation and the IPS line cell culture variation. And, we need efficient strategies for increasing sample size to strengthen the conclusions.
In addition to including multiple individuals in a pool, the sort of conundrums or challenges you just described, does it help to have replicate lines from individuals in a village to say, does the same person always act the same way in a dish of 20 other people or donors?
Yeah, I mean, in general, if you can do replication, that's helpful. I think
It's
we
just,
could,
yeah.
as a group, talk about like the experimental design strategies and having anchors of the same individual between multiple batches and pools, um, as well as different clones for the same individual if we want to tease apart variation between clones and individual. But, for us, we did a whole experiment. We did all 17 individuals in different replicate experiments so we could look at, the correlation between the two. And that was very helpful. I think there's probably some stochasticity and the starting number of cells matters. I think we probably, yeah, I think the starting number of cells matters. If you have, very tiny experiments like micro wells or something, you'll have, well, I guess we'll find out as a field if someone has, can overcome this stochasticity. But yeah,
Well, can I make a comment there about starting number of cells? So
yeah,
in the gastroloid world, the starting number of cells appears to be really critical.
yeah,
And if we're thinking about morphogenesis and morphogens, the starting number of cells is going to influence the signaling environment where the morphogens are acting. So I just wanted to make that comment. It's not just us that's worrying or interested in the starting number of cells.
yeah. And I think about that a lot because I would love to do pooled culture with some of these new gastroloid and embryoid technologies. But in those cases, they have a critical cell number that is quite small that would limit the number of individuals you could start with. For these organoids, we're typically starting with 10,000 cells. And in our case, we did the pooled culture for the first 16 days in 2D, and then we cryopreserved. So there is still this kind of dissociation and remixing when we seed. So we get to kind of start over with the seeding. But if we had uh, gastrolite, and I'm not fresh on the number, but if we were starting with like 200 cells in a micro well or something, I think that
Yeah.
would be prohibitive to have consistent mixing of individuals per a line. I think you could have a kind of statistical mixing, and then you'd have to barcode the gastroloid of origin from your analysis, and you could find, the number of cells from each individual for each gastroloid to
Yeah.
know whether there was mixing. But I think that would be an extra level of experimental design challenge because
Yeah.
a 50-50 mix might be different from a 95 to 5 mix in terms of the donor versus host. Though one of the lessons I didn't really talk about here is that most of the gene expression divergence, nearly all of it, seems to be cell intrinsic. And so you may not have to worry as much about cell extrinsic effects when you're at, say, 95 versus 5% of host versus donor. That most of the gene expression variation that we would like to explain, in our case across species, but in other studies, even within a species, seems to be cell extrinsic and would be amenable to genetic mapping without worrying as much about the overall composition. That being
In,
said, some lines respond differently to patterning factors than others, and you might have composition effects, which is why we wanted to look closely at the composition of pool types.
when you were discussing this in response to Carlo's um, questioning, you made this comment, which I think deserves our attention a little bit more, which is that the dopamine system is different from the cortex. And I wanted to say a comment of my own experience was that when we first started trying to grow dopamine neuron precursors, we found it very difficult to grow. And the dopamine neurons come from the floor plate, and the expansion of the floor plate is highly constrained. And so I think that in one of the big differences between using this to study dopamine neurons and the cortex is that in the cortex you have these intermediate precursors which are designed to expand, where you could find differences. Actually want to say that that's potentially extremely interesting. If the variation, let's say, between humans is not in the dopamine system so much, but in these later stages, as the Arlotta lab and others suggest, that could have biological meaning.
Yeah,
Yeah,
and I think that also highlights why the cortical pooled culture might be harder because you
Mm.
have all of this, intermediate progenitors and secondary expansion and prolonged period of expansion that can
right,
compound these subtle differences.
right.
And we have less of that here. But you know, even though these are constrained and phylogenetically older regions and cell types, we think that they are a homologous cell type that is living in a new environment of our large brain cellular ecology where their role has changed in terms of their connectivity demands and bioenergetic demands. So we think it might still be a locus for consequential evolutionary changes governing connectivity during development and this buffering. And we found some evidence for that in this first study establishing the system.
No, I don't think there's any question. I think that
Yeah.
I would just want to nail down the first part of your experiments a little in our discussion a little bit more,
Yeah.
and then that sort of sets us up to ask about the meaning of the oxidative stress or stress responses. That strikes me as super interesting, but really quite complex, isn't it? I mean, I don't know. I mean, I was overwhelmed by your paper, to be quite honest. I mean, it's just this substream of studies, and when at the end you start talking about rotenone, well, let's talk about that. Is rotenone the right stressor? Is there? I mean, rotenone is physiologically perhaps or clinically relevant, but talk to us a little bit about that. Is there are there many ways in which rotenone is toxic, or is it well understood?
Yeah, so rotenone disrupts the electron transport chain at and that will lead to bioenergetic stress, including oxidative stress. At high levels, rotenone can also disrupt other cellular processes, like microtubules. And we selected this assay because we had a starting point to work from these disease studies. where we knew relevant concentrations. In the lab, we've also been trying to use piracidin and hydrogen peroxide as maybe more focal
Mm.
disruptions. But this is an assay that can elicit known oxidative stress response genes that we thought would be sufficient as a first pass for unmasking additional species differences. Um, if that answers the question.
But
And
I mean, there's no real answer really. And when
Yeah.
you talk about mitochondria and electron transport chain, one of the earlier on in your story, you talk about mitochondrial transport being a particular along axons, being a particular sort of cell biological source of difference in the different species. Could also be perhaps, I mean, interesting to think about that as an assay system for differences between the different species you look at.
yeah, you showed some assembloid data. Did you do any comparison about how efficiently a chimp's versus human dopamine midbrain organoid innervates a forebrain organoid?
When we started the project, we had this hope that we could ask the question about cell intrinsic versus, or species intrinsic versus extrinsic factors regulating connectivity, because we could toggle the source species and the target neurons. But we found pretty quickly that the variability in the size of the source and target organoids would kind of dominate this, and it was difficult to quantify. And I
Gotcha.
think this relates to Daniel's question in the thread. How could you look at connectivity in these organoids and at what stage? And initially, we wanted to use these assembloids, but we think they lack the consistency, and our hypothesis is for cell intrinsic differences. And so, what I would love to do, but we haven't started yet, is build on the great kind of xenotransplantation studies that have been used for the cell therapy validation, and to where you can even recapitulate some of the known connectivity patterns if you transplant into midbrain, and to transplant a pool, a mixed species pool of these dopaminergic neurons that are labeled, and then quantify in this more physiological and common extrinsic environment the connectivity differences. So, to answer your question, we thought about doing it in organoids, and we could probably build a more consistent system with like a mixed species dopaminergic organoid with different colors where the size is controlled and the quantity is controlled, and then single hosts of different hosts, and kind of do a connectivity development comparison. But we
Yeah, hard to
thought
get it
that it would
comparable.
be we thought there'd be a lot of variability and it would be a huge investment. And we had our hands full with the genomic data. There were a lot of advances in there to just do fair comparisons of the gene expression and chromatin accessibility between species. Janelle, who's on the call, wrote this whole extra tool called CrossSpeak for finding the orthologous non-coding loci with base pair precision for fair quantitative comparison. So we had our hands full just with the genomic and molecular data at that point.
Hi Alex. Great talk. Really, really great. Your techniques are amazing. Your tools are super interesting. Could I ask a question that might involve going back to a slide?
Sure.
So the slide that you showed, the proportion of across the different species where it was day 40 and day 80, very early on, you show
Yeah.
sort of a difference that seems like the human cells have far fewer cells in the organoids by day 80, and it seems sort of like universal.
Oh, let's take a look at that.
Yeah. Because
I
initially
think
my initial thought was, oh wow, maybe they are having a different response to oxidative stress. They're not surviving as well, which is the exact opposite of what you're showing with sort of the gene expression. But so then I was wondering, like, are the cells proliferating differently? Like, are they out surviving or are they, differentiate or proliferating at greater extent? So here you can see it seems like the human populations are reduced by day 80, compared to day 40.
Mm.
So is this because of de cell death or differences in proliferation rate? Um, and would you see the same proportional changes if you look at species only organoids? Um,
Yeah, I think this plot here is a mixture of the within and the between species. And if you look in the supplement, we have the interspecies and intraspecies broken out separately. So
okay.
this plot here reflects what we're looking at here.
Hmm.
But when we break it out separately, I don't actually think there's a difference in the survival of the human dopamine neurons at these later stages. I think that this reflects some differences in the sampling between species. So I we did not see strong statistical evidence for differential survival. It was something we definitely thought about and looked for in the beginning of the experiment. Um, so yeah, I agree. It looks like that here, but maybe we can pull up the supplement and take a closer look at the
No, no,
panels
no.
we
It's
see.
I take your word. This is the other question I was going to
I
ask.
actually have it here if you'd
Oh,
like
okay.
to
Yeah, go for
see.
it. Yeah, why don't you walk
Awesome.
I,
us through it? That'd be great.
yeah, I can share my screen.
Wonderful.
Um, okay, so I think this panel G is probably what you're looking for. So there were actually three separate experiments done. Actually, I guess this is still not broken down to inter and intra species, but this is broken down into the three different experiments. And I think the one that Alex showed was all of the data combined. And there was one experiment, I think Alex mentioned it very briefly, that we call this replicate experiment, where this kind of really shows how important the health of the stem cells going into the experiment are, because in this case, it just so happened that the chimp lines were healthier than the human lines to start out with. And I think this also kind of had to do with the processing when Sarah was seeding the lines. And so
Oh yeah,
when
yeah.
we,
This initial replicate
yeah.
experiment, all the human lines were dissociated first, which you
Right. So
never,
they were kind
you
of
know.
just sitting around longer. So then when we redid it, Sarah and I did it together. And so we made sure that like all of the lines were sitting dissociated for the exact same amount of time. And
This,
that's what we're
when you
calling the
say
first experiment here.
dissociated first, you mean from pluripotency before you mix them.
Yes.
Yeah,
That's the
exactly.
sensitive spot.
Yeah,
Yeah,
it's really
exactly.
sensitive if you want to count every line and seed them in equal numbers to start, which we thought was important. I think, we've learned a lot from doing these experiments, and I think you could get by with something called like packed cell volume, or you could have a counter that lets you count like 8 or 16 samples at the same time. And then that is the time lag in any of these pooled culture experiments, is the time dissociated when you're counting cells. And if you're counting in serial, some lines are dissociated for longer than others, and that needs to be randomized across species. And we found that out the hard way in this replicate experiment. The majority of our data is not from this experiment, but from this one. But we could still use this replicate experiment to show even under these conditions, the gene expression is highly
Mm.
correlated between individuals and cells and samples. So
Yeah, and in all of the differential expression and accessibility analysis, like we use random effects variables to account for which experiment the cells came from.
Nice. So the
Mm.
follow-up, sorry, is how do you, I mean, so there's going to be maybe differences in signaling cascades and temporal aspects to sort of optimizing by species these protocols. So can you speak to, how you determined, the end protocol, whether or not it sort of benefited one species over the other, or whether, you find huge differences when you shift things slightly across species? So, like the protocol might sort of might be optimal for one species over the other, and yeah, I'm just curious how that, you align those factors and sort of compare those to get to one protocol, if that makes sense.
yeah, so we started kind of naturally by using the protocol Sarah had developed, towards clinical trials in humans and applying it across species. And we had to tweak the density of cells for each species. We grew all the cells in this common media, stem cell media that Sarah had used previously. And we didn't find the need to tweak it that much. The orangutan line that we used, this one line, tended to be a bit more rostral for a given amount of wind stimulation.
Mm.
But besides that, we saw pretty similar compositions between individuals. I would say that the mixed species organoids tended to have a little bit higher on target. um, there's this idea that the, patterning of some lines can help other lines, or the health of some can help others.
Hmm.
And we saw that recapitulated in some of our pilot experiments. The other sort of surprising thing was that we got the same off-target regions, or not off-target, we're making ventral midbrain, but we got the same heterogeneity with human and chimpanzee. But the rhesus macaque in black here was very high purity for just the dopaminergic lineage. And for whatever reason, the rhesus macaque, all three lines, responded even better to this protocol developed for human
Mm.
than across many human lines.
Yeah,
Um,
the fact that they sort of differentiated quicker, got to the place of producing astrocytes was why I was wondering if the protocol was actually maybe like benefiting it differently, or changing, the sort of rate of yeah,
I
in
think
it.
that points to a real challenge for how far phylogenetically you could expand
Mm.
interspecies organoids, and when you get to more extreme differences in timing, if it would overwhelm the system. Like, I don't think this would work for human and mouse culture. And
Right.
Junwoo has even shown some evidence for competition between human and mouse cells, but did not see evidence for human and rhesus macaque competition, and nor do we in the culture model.
Okay. Thank you.
Yeah, you bet.
There's a couple other questions in the chat. Sridevi, would you like me to read your question, or do you want to just jump in? So I can read it if she's still muted. Her question is, would you say that oxidative stress genes you identified in human dopaminergic cells are more unusual than in the cortical organoids, for example? Unusual, I don't know. Sridevi, what do you mean by unusual there? I don't, maybe she can't talk.
We haven't done the one-to-one experiment with cortical organoids here. We were actually really excited to see that this catalase gene that was,
Mm.
very significantly quantitatively upregulated in human compared with non-human primate matched what was reported in human cortical neurons. So in that case, it might not have been a dopaminergic neuron specific, but a more general feature of human neurons where there's orthogonal in vivo tissue level data. Um, and, this follow-up, how do these differences map onto ideas about protein turnover rates and metabolism that might drive species differences in developmental tempo? I think that's a really interesting question. I think that the differences in developmental tempo are going to be much more subtle between human and chimpanzee. And we are able to use pseudotime as a covariate in the experiment. So, I think what we're seeing is independent from developmental tempo, but it is interesting for any stimulus-dependent response experiment, whether when you're comparing between species, whether you would harvesting at the same, you know, absolute time between species might lead to differences due to like, subtle differences in protein turnover, or metabolism, or RNA turnover. Um, but
Yeah, I'm
I
also
think
curious,
those.
I guess.
Yeah.
I was
Oh, go ahead.
thinking like similar experiments in zebrafish, right? Like with temperature differences. I think this is from Jay Shandur's lab like a while ago, the changing temperature to change embryo development in zebrafish, like how that changes cell type composition, and even within the same tissue, like different cell types mature at different rates when you do that. Yeah, like just given that you have similar sort of stress response, oxidative stress response related genes, I wonder if subtle changes in that sort of overall physiology can change your results.
Yeah, I think it's a really interesting question. I think it'd be hard to fully assay, but I don't expect those differences to be so great between human and
Mm-hmm.
chimpanzee. For a lot of these timing differences, we try to account for it by just sampling multiple time points. And we can do that across developmental time as well as across the stimulus-dependent responses. And when we see consistency at those multiple time points, it argues against the, developmental tempo or protein turnover as the explanation. But if you have really temporally specific species differences, then it's a little harder to rule out by the multiple time point sampling method. And for that, we've tried to use pseudotime as a covariate, but I could see some cases where that might not be sufficient.
We got another question from Soki. Do you want to jump in, Soki?
Okay, yes. Alex, I think it really relates with our previous discussion. My question is about whether it's this differential response to oxidative stress is actually related to the maturation state, right? So, because human dopaminergic neurons are mature little slower to the macaque. That's what you are saying, right? I
Yeah,
think.
in this case, we compared human to chimpanzee, and we found minimal differences in maturation, or to the extent that they're subtle. And there's a distribution of cells along this maturation trajectory such that you could account for it in the differential expression comparisons. So, while I agree that there might be maturation specific responses, that's not what would account for the differences in the way our experiment is designed and with the maturation stages that we've observed.
Okay, so that was actually between the human and chimpanzee.
Yeah, that's right. I think it would be harder to do that comparison with human and
Macaque.
rhesus macaque for
Yeah, that's the
And the
bigger
fully
time
maturation
mature,
difference
stage.
they showed us, right?
okay, the fully matured stage that you tested
Sorry, say again?
for the oxidative stress.
Yeah.
When did you actually do the
We did
uh,
that at day 80 of
the
differentiation.
Mhm.
Yeah. And we saw similar distributions of human and chimpanzee along pseudotime for that stage. But we can also assign every cell a score and account for that. So comparing cells to the same maturation stage.
Mhm.
Yeah, because the big gap you showed us in timing was the macaque at an early time point, right? That had jumped
Yeah.
forward.
Yeah. And I think that, I view that
Mhm.
as like, you know, positive control evidence that, our system can account for maturation.
Mm.
And we have, orthogonal neurogenesis to gliogenesis switches. But the human chimp differences are much more subtle. I know that, there's been some evidence for these differences in cortex, but even there, when we've analyzed the data, it looks quite subtle. Um, but it's also something you can account for in your comparisons.
Related to this, we have another comment that says, also, I think NPC early neurons are more sensitive to oxidative stress. So it seems it's going in the opposite direction. If human cells are less mature, we'd expect them to be more sensitive, maybe. So,
Yeah.
but again, that should be taken into account, right? Because you
Yeah.
have the pseudotime correction.
Yeah, we have used the pseudotime correction for, for additional analyses. Janelle has made like 50 models at various points. So,
50 of the pseudotime.
or just
Or
of the whole
of
experimental design, in
what
finding the right
covariates to
dreamlet
put in there.
model and the pseudobulk bins and everything. She's really explored linear mixed models deeply.
Could I ask us to think a little bit about transplantation and where the clinical field is at the moment? And particularly because Nova have just stopped all their cell therapy programs. There's clearly, the reason they do that is because we're in the sort of business end of things, which we've got to take seriously. They sort of view cell therapy as being altogether too expensive. So I have two questions really, which are sort of related to this. One is rather precisely focused on these neuron subtypes that we've been discussing, which is, do you anticipate that we're going to make really significant progress getting different kinds of dopamine neurons under control? in this system, so that we're talking not just generically about dopamine neurons, but making
Subtypes.
specific subtypes.
Yeah, so for that question, the Studer Lab has this Boost Plus protocol where they present some evidence for substantia nigra versus VTA, but it's just based on a few marker genes. And at early stages of development, these marker genes can be more promiscuous. One of the things that we've seen in our more extensive analysis of cortex and looking at the actual fetal developmental samples in brain atlases is that there's this big gulf between the prenatal cell type diversity and the adult cell type diversity. There's a lot of room for post-mitotic fate refinement, and I think that is occurring at later stages in a circuit context. And maybe, like the Suter protocol, you can do some things that will bias the acquisition towards more substantia nigra, but I don't think you really know until you do. I think you need to mature these further in a vascularized environment. And the best environment for that right now is, transplantation, in a rodent model, for example. And I think we need some post hoc single nuke RNA-seq there to determine the extent to which we capture these different subtypes. And it's
Yeah,
not just substantia nigra. There's like 6 or 7 subtypes within
sure,
substantia nigra in the adult. And,
sure.
in my view, there may be some, it's exciting that, there's a step forwards in this from the Studer Lab, but I think we're still a long way from getting to something that would purely result in substantia nigra and even biasing towards the vulnerable subtypes within there. My guess is you get a mixture of these post-transplants and that the environments can influence some of this post-mitotic fate refinement. And, part of that I think is just that these, even if you looked in third trimester fetal samples, I don't think you would see these subtypes. I don't know that it's all so much as an inherent limitation of the IPS system as it is that the fate
Yeah, yeah.
refinement has not yet occurred at these developmental stages. So we're looking for signals at earlier stages than we would see strong signals in vivo.
No, that's very helpful. I wonder also whether we might just think more sort of practically about this kind of technology being used in a clinical context. Can you imagine storing the cells at many different stages of their maturation and really testing them out as for their transplantation efficacy? Is that possible? Right now, we transplant cells that are really quite immature, isn't that correct?
Yeah,
And that's an old result.
my understanding is that the transplanting more mature post-mitotic neurons, these are projection neurons are just too sensitive for that procedure, so that you have to transplant at these immature stages
Right.
for the cells to survive.
But that result is, I mean, that's a 20-year-old result.
Yeah,
If
I,
it,
I imagine we could optimize for transplant survival or add some neuroprotective, transgene or
right,
something for
exactly.
short
Yeah,
duration
yeah. I
to
mean, if you were
prevent cell
really in
death.
the
Yeah.
business of transplantation, that might be a topic we would want to think through a bit more. Anyway, thanks. I just thought
I
it would
think
be
that would
useful
be,
to think
you could
through.
imagine even pool, multiplexed experiments to, try to address, discover factors that would promote survival in your
Yeah,
transplantation.
right, exactly.
Um, so, yeah, I would love to
Yeah,
do
to
more of those
co-transplant.
experiments.
Something else, I don't know really what data or information we have to dig in deeper, but something I don't think is often thought about is we all very much think about differences in the efficiency of different human lines to produce dopamine neurons. But once we're getting to where we are now, talking about putting them into different people's heads, is there something we could look at or understand about the person we're putting the cells into that would give us information or um, similarly to putting a transplant in with a growth factor that'll help it do whatever it needs to do once it gets there? We're kind of at this point thinking everybody's head is the same. You stick cells in there and they'll behave just the same as putting them into the next person's head. But formally speaking, the variation could be just as big as the cells coming in as to if there's a little bit of a although you're putting into an organism that was formed in a balanced, normal way. I guess,
Yeah, I
is
mean, in
there
other
ways we can
fields, folks think
understand?
about the immunological context, for that and modifying the MHCs. In Parkinson's, I think folks think a lot about the, alpha-synuclein or other protein pathologies that might affect the transplant cells. And I think the argument is, that these cells can still go 40 years or something,
Right.
in resisting this. But on the other hand, the burden of protein aggregates may be so much greater now. So you could imagine, second and third lines of defense based on the protein pathology or something else, as well as just these survival factors that Ron alluded to that might allow
Hmm.
more mature projection neurons to survive. And then the other point is, can you get the neurons to mature more quickly post-transplant? Can you modify some of the maturation and change the metabolism and tweak it in some way so that they take on a more active role more quickly. Um,
A lot of dials to, very complicated dials to tune.
yeah, but we started this with a interest in the evolutionary questions, but it certainly made me more interested in the cell therapy questions and in using a data set like this with the multi-omic data, we can do these in silico perturbations and predict gene perturbations that might promote cell
Mm.
health or be neuroprotective. And, that falls outside of the narrative of, this first study we're presenting, but it is another application of the resource to do these in silico perturbations and identify. And we can identify known treatments like, the in silico perturbations would say like PBX1 is a neuroprotective gene regulatory network and some of these other ones. I don't know off the top of my head, like BOC2 or something, maybe more related to the oxidative stress. But it is, I think, a useful resource for mining neuroprotective factors, even with the human data alone.
Right, right, right.
Albeit these are still immature stages of development compared with the adult brain, but the bioenergetic stress response does mimic some of the oxidative stress and other stressors that occur with aging. So it is proxy for some of the experiences that a more mature neuron may respond to.
This is a lot of fun. I was thinking, you mentioned Jun Wu earlier,
Yeah,
and I was thinking about his experiment where he gives a rat a mouse forebrain,
yeah,
or the other way around. I can't remember which way around he did it,
yeah,
but anyway. And I think he gives a mouse a rat forebrain. I think what he does because he has a genetically, he has a
yeah.
FSF2 deficient mouse or something. But I was thinking you could imagine testing out these different dopamine neurons in where you actually replace the dopamine neurons in the animal you're interested in. Let's start with a mouse, but in principle, it would be nice to do this in a primate. And so you actually have a primate wandering around with dopamine neurons that come from different species. That would be an experiment that should teach you a lot about the behavior if you were really interested in the behavioral consequences.
Yeah, I've always been concerned about any experiment where you're mixing human and animal material. You have to be concerned about the ethics and public trust in
Sure.
science. And I think the most concerning, not so much organoids as, human material outside of an organism, but are these chimeric experiments. If you were to give a monkey a human cortex, that would trigger,
Mm.
serious alarm bells and would be difficult motivation,
Yes, but we're
to
just talking at the
justify.
present time. We're
But
just
but I
chatting.
think, a few 100,000 dopaminergic neurons would have much more therapeutic relevance and would trigger less ethical scrutiny. It's not the seat of consciousness, but it might affect like social affiliative behavior or working memory or other things that we can analyze. And it would help us to, it might even be a source of neurons for transplanting. So, I would, before I were to do an experiment like that, even in rodents, I'd want to really work with a bioethicist on it. But I think you could make a strong case for the therapeutic relevance and not endowing a animal with additional capacity for suffering or, betraying the dignity of human samples or something. But it would take a bioethics argument first. I think, we thought about the xenotransplantation as an even smaller number of neurons, but yeah, it is only a few 100,000 to begin with. So it might, I do think it's a very interesting experiment. These embryo complementation experiments where like the whole LMX1A lineage is mutant in the animal and such that it can only grow up with the human donor cells would be a interesting
Yes,
way of studying
yes,
the function of
yes.
these en masse.
I mean, I take it we would do this thing with care and thought, but let's just imagine the outcome of an experiment like that. It would be extremely interesting to ask what would, let's say we don't start with a human neuron if we're too worried about humans, but we start with this neuron of another species and we test it out in an environment where it can actually make all the connections it's supposed to make. It would teach us a great deal about what dopamine neurons are doing, how they pull this off. They're some of the first neurons to be born in the forebrain, in the brain which is anterior to the hindbrain. And they as soon as they become postmitotic, they're producing dopamine. So from the very get-go, you have this cell type, which is a controlling cell type that comes from an organizer that's producing its neurotransmitter. There's a
Right at
whole
the beginning.
series of questions here that are, I think, of great interest. Anyway, thanks so much for talking
Well,
to
that
us
would be
at
so
such length.
exciting. If you have a way of, making maybe the smaller version of that happen first, we should talk more. I'm definitely interested. I think we need a in vivo context for, certainly for studying any impacts on behavior, but even for studying for really studying the connectivity differences. And I think to get to the cell type specification, we will need an in vivo model. I don't think maybe we'll get to substantia nigra, but not to all these diverse subtypes within there in vitro. I hope someone proves that wrong, but there's been a lot of investment in these protocols, and I haven't seen
Mm.
strong evidence for those subtypes.
Do you feel like we
Cool.
have a good handle on the characterization of the types we're looking for already? Or do we need even more of that before we know what we're looking for?
Um, it's a small population, and these adult brain atlases have focused more on cortex. I think the Evan McCosco paper is beautiful from 2022, and they give a first pass of just substantia nigra. It doesn't have VTA. They do multiple species, which is exciting. You need some strategy for enriching for the dopaminergic neurons because they're rare, or you just need like much more low cost, single cell sampling to get sufficient numbers. But, why they're so exciting from this trade-off is also why it's so hard to capture them in sufficient numbers for a cell atlas. So yeah, I think there could still be more diversity that has been unidentified. I mean, certainly there is because the VTA. I think there's some good mouse studies where you can see this, but in terms of human and non-human primate, there's still limited data.
Yeah.
And,
Shouldn't
Evan's
Even
study
know what we're
suggested at
looking
least
for.
one
Yeah.
primate-specific population already. So,
And
we
that
be
came out of the
talking about?
McCosco paper?
yeah,
Sorry.
comma 2022. Yeah.
I was just thinking along these lines of very specific features of the dopamine system that are sources of stress. We should at least put the dopamine transporters on people's agenda. The place where, at least as far as I understand it, the place where the dopamine neuron is really sensitive is where it releases dopamine, and then it very rapidly takes it back in again. And it's that cytosolic dopamine before it's repackaged,
Yeah.
which is the dangerous dopamine.
Yeah.
And of course, that whole cycle and the timing of that cycle is all to do with the way dopamine acts as a reinforcer. So I would think that the dopamine transporter system might be something. I mean, if we were really going to pursue this sort of thinking, it would be great to sort of build that into your experimental design.
Yeah, I mean, we'd love to do follow-ups. I'd love to talk more how we might do that, or if, maybe we need more comparisons in adult human and non-human primates, and, samples versus just the
Yeah, it's
organoids.
very stimulating, Alex. I mean, I don't even really know the population genetics of the dopamine transporter genes in humans, which I should go and look at.
Yeah.
Yeah, great.
All right, well,
Thanks a
thanks
lot.
so many folks for staying after for this question session. I wanted to leave time, and I'm glad we had extra time here.
Yeah,
Thanks so
thank you
much.
very much.
You bet. I'm also happy to follow up if you want to
Yeah.
speak more.
Excellent. And next month we have Michael Wells is coming back. He gave a talk, I think it wasn't even a year ago, but they have another village preprint out, and he'll be talking about that next month. So we can jump right back into pooled culture.
All right,
Thanks
thanks
so
everybody.
much.
Great to see you.
Cheers,
Bye-bye.
guys.
It was great to meet everyone. Thank you.
Likewise.